Welcome to Chapter 16 lecture, where we will focus on blood. Now we're just going to cover about half of this chapter, so we're not going to cover the entire thing in this one video lecture. Here's the outline for Chapter 16. This video is going to cover the plasma and cellular elements of blood. We'll go into details about how blood cells are produced, or light detail I should say, and then we will talk about red blood cells and finish the video on some disorders associated with red blood cells. In class, we will pick up with platelets and their role in hemostasis and coagulation. Here are the learning objectives for this chapter. Notice that I've made some changes to these learning objectives, including the first one I just underlined there that we're not actually covering. And then the ones in red are changes that these are learning objectives that I've modified slightly to reflect the information that that is contained within the chapter material. In addition, the very last learning objective is not in the book, and it's one that I created because the book and my presentation will go over medications that are used to prevent blood clots, so I thought I'd put one in there. But this video, let me go back, this video is really just going to cover learning objective 16.1.1 through 16.3.4, and then we will cover these learning objectives in class. Okay, so let's talk about blood. So most of you, when you think of blood, you think of red, you think of red blood cells maybe, but the blood is a very dynamic, very complicated element to our body. And before I go into the elements of it, just as a reminder, you may or may not be aware of the total blood volume of an adult, but it's always fun to know. So for adults, if you have, I think it's around a 58 kilogram female, then you're going to have roughly about 4 liters of blood. For men, an average 70 kilogram male is going to have a little bit more blood, and that's about 5 liters. Okay, because blood is, the amount of blood is associated with one's weight, so the more you weigh, typically the more blood you're going to contain. These are just averages, so but just generally it's considered that we have about 5 liters of blood in an adult. All right, so let's talk about the components of blood. Blood is divided into, the elements of blood are divided into 2 major categories. The first one being here plasma. You may have heard or associated the plasma with the watery elements of the blood, and that's true, but I think that term is a little misleading because plasma contains way more than just water. Okay, so yes, vast majority of plasma is the water, but within the plasma you're going to have ions, including sodium and calcium and various other ones. You're going to have a wide variety of organic molecules, mostly nutrients, lipids, proteins, glucose. You're going to have a wide variety of proteins, including albumins, globulins, fibrinogen, transferrin. All those we're going to cover in this lecture. You're also going to have some waste products, urea and nitrogenous waste. The plasma also contains trace elements and vitamins, so vitamins and minerals, including iron. And then, as we've discussed in previous lectures, how blood is and the circulatory system is critical for the distribution and transport of gases, including oxygen and carbon dioxide. All right, so that's the plasma elements. If it's not plasma, it's these cellular elements. And there are 3 categories of cellular elements found in blood. The first one are red blood cells. There are only red blood cells in this category, specialized transporters of gases. The second category of cells found within the blood are white blood cells, or leukocytes. And there are 5 different types of white blood cells that are shown here. Lymphocytes, including B and T cells, monocytes, neutrophils, eosinophils, basophils. These all have very specific functions for the immune system and defending against foreign pathogens. We're not going into details in this chapter. The third type of cellular element found within blood are platelets, and these are little tiny fragments of megakaryocytes, and they are critical for the process of hemostasis, which is preventing blood loss. Okay, so those are the plasma and cellular elements of blood. Let's just see if a couple extra points to emphasize in regards to the plasma. It is considered the extracellular matrix to the blood, and it is, like I said earlier, a majority of water, and then that remaining 8% is a combination of the various proteins that I just went over, and 1% dissolved organic substances. The last thing I want to emphasize on plasma is that it is very similar in features as the interstitial fluid, the fluid between cells. However, plasma, there are a few key differences that really play a big role in homeostasis and fluid exchange for the most part. So even though it's 8%, the plasma contains more proteins than the interstitial fluid. The albumins, the globulins, fibrinogen, transferrin. The presence of the protein in the plasma, it increases the osmotic pressure. Remember that term that we covered in the last chapter, the colloid osmotic pressure. Whoops, I don't know why I put M there. Colloid osmotic pressure. It's the pressure due to the presence of proteins in the plasma. So plasma has a higher osmotic pressure than the interstitial fluid, and that results in water leaving the interstitial fluid and being absorbed back into the venous end of a capillary. So it's really important for capillary function and fluid exchange, regulation of fluid exchange. Okay, so extra information on red blood, or the cellular elements, I should say. So red blood cells, the technical term is an erythrocyte. Okay, and just a heads up, red blood cells, they lack several organelles, including a nucleus, but also some other ones. And that's an important bit of information to know because that really dictates their function. And as I mentioned, there are those specialized transporters for oxygen and carbon dioxide. Nothing too surprising there. White blood cells, they are called leukocytes, not erythrocytes, leukocytes. They are what I'm calling the only functional cells in in the blood, not our body. And that is mostly because all leukocytes contain one, sometimes more, nuclei. But all in all, they're very critical for immune function and defense against pathogens. I've covered the different types of white blood cells previously, so again, we're not going into the functions here, as we will later in the semester when we cover the immune system. The last group of cells that are found within blood, you're probably the least familiar with, platelets. Technical term are thrombocytes. Okay, these are critical for hemostasis, or blood clotting, the reducing blood loss in case of damage. Just like a red blood cell, they also lack a nucleus, and that's because these platelets are technically fragments of a much larger cell called a megakaryocyte. Again, we'll cover that in class. However, most of us are familiar with impacts to our platelet count, or number of thrombocytes. So if you've ever taken an NSAID, a non-steroidal anti-inflammatory drug, okay, Advil, Motrin, Aleve, etc. These NSAIDs, they are all cyclooxygenase 1 inhibitors, or COX-1 inhibitors. And this is an enzyme, and its major function is to block prostaglandin production, which if you reduce prostaglandin, you can reduce fever, you can reduce the pain sensation, and reduce inflammation that's associated with injury, etc. So that's what the NSAIDs do. However, these COX-1 inhibitors also because they knock out this critical enzyme in platelets, it reduces platelet count. Platelets actually die. And it takes a day or two for our body to make up. If you just take one dose of an NSAID, it takes a day or two for your body to make up for the loss of platelets. So that's why those who are on NSAIDs could be at an increased risk of bleeding, but I'm not trying to say that these NSAIDs are going to cause you to bleed out. Okay, I'm just saying that they're technically a form of blood thinner. All right, so let's talk about blood cell production very quickly. We're not going to go into depth here. So all blood cells, regardless of red, white, or platelets, they develop from what is called hematopoietic stem cells, or HSCs for short. These are pluripotent stem cells, and what that means is that the HSCs, they can only produce red blood cells, white blood cells, or platelets. They cannot produce non-blood cells. They can't produce skeletal muscle cells, they can't produce neurons, etc. The process of producing these various blood cells, that is hematopoiesis, okay? The synthesis of all blood cells. This occurs in our bone marrow, it occurs during embryonic development, it occurs in what I call postnatal environments, so after you're born and actually until you die, we're constantly producing red blood cells, white blood cells, and platelets, hopefully. Good news is we're not covering hematopoiesis in depth, which is figure 16.5, nor are we covering the cytokines that are involved and regulate those. That can be found on page 515, so don't worry about that. I just want you to be familiar with the source of red blood cell production and the process, but not the details of the process. Instead, I want to focus more of our time on understanding what a complete blood count, or CBC, is. So this is an analysis, a clinical analysis of blood components, and it's going to compare, the goal here is to compare a sample of one's blood to what we call a quote-unquote normal range. However, CBCs, very easy, very cheap, cheapish, I should say, diagnostic tool, but it can be a very valuable indicator of many health conditions. It provides very quick insight to physicians and their teams on what's possibly going on with a patient. So this is the graph from your book that's trying to explain what a CBC looks like. Okay, so you go into the clinic. Most of us have had a blood draw. Many of us have probably had a CBC done, whether we know it or not. But go into the clinic, they take a blood sample, usually 5 to 10 mils, and the very first thing they do is they take some of that blood and they put it in a tube and they spin it down in something called a centrifuge. And when you spin down blood, you're going to result in a tube that looks like this. It results in what we call packed red blood cells. And this is an indicator of your hematocrit. Okay? So your hematocrit is basically the percentage of red blood cells compared to the total blood volume. Okay? And based on this, if a hematocrit is too low or too high, that indicates that the viscosity of your blood is abnormal, and that can have various problems can be associated with either thick or too thin of blood. Okay, so that's what a hematocrit is, percentage of red blood cells compared to total blood volume. The other information, not just by the centrifuging method, but they're going to take a small sample and then look for the amount of hemoglobin that's found within the blood. And this is indicated, it says right here, grams per deciliter, so grams of hemoglobin per deciliter. So this is literally how much hemoglobin is found within your blood. Related to that, CBC will also give you a total red cell count. So this is just a number of red blood cells. Okay? So really these top three numbers are giving you some indicator of relative RBC levels and health. If it's not an RBC or not information about RBC, then it's going to give you some information about white blood cells. And I drew that a little incorrectly here. Okay. So this is what a total white cell count and differential count are. So a total white cell count is exactly how it sounds, just the total number of white blood cells that are found within your blood. But that doesn't give you information about the specific types of white blood cells. It doesn't differentiate between them. So that's what a differential count is. It actually assesses what's the number of neutrophils, number of eosinophils, number of basophils, number of lymphocytes, number of monocytes. Okay. And so that'll give you an indication of your general white blood cell levels and health. And then the last part of a CBC is going to look at your platelets or your thrombocytes, and this is an indicator of your body's ability to clot. So if it's low, you may have problems trying to clot your blood. One thing I'll mention here is that what's shown on the right side, males versus females, these are normal values or normal ranges. And I do expect you to be familiar with the normal ranges of both males and females for a CBC. Okay, the last thing on a CBC, it may or may not, but more often than not, it does include some additional information up here. The MCV, the MCH, and the mean, and the MCHC. This is basically looking at the average volume of one red blood cell. Okay, so if it's small or too big, that's useful to know. How much hemoglobin is contained per red blood cell. The hemoglobin number down here was in a whole blood. This is estimating how much hemoglobin is in one tiny little red blood cell. And then the MCHC is the amount of hemoglobin per volume of one red blood cell. So it's really the ratio of the MCV and the MCH. All right, so that's the CBC, really useful clinical diagnostic tool. Let's talk about red blood cells in a little bit more depth, and then we'll finish it up with some anemia and problems associated with red blood cells. So here's just a cartoon and scanning electron micrograph image showing a typical red blood cell, described as the term used to describe them biconcave. Okay. So concave shape, it's going to have an indentation shown right here. So it's not just flattened, it's not flat, it's actually kind of pushed towards the interior or towards the middle of the cell. Bi means two, so it's actually both sides of a red blood cell. They're kind of smushed towards the middle of the cell. They're not round, little ball-shaped cells like many of the white blood cells. Okay. Again, notice the lack of nucleus. You can't see, or they didn't indicate a nucleus on within the red blood cell because they don't contain them. They are the most abundant cell in our blood, as you probably ascertained from when we were going through the CBC information. However, to put this into context, for every microliter of blood, microliter being a tiny, tiny amount, you have an estimated or roughly 5 million red blood cells, and this is compared to just roughly 4,000 to 11,000 leukocytes or white blood cells, or roughly 15,000 to 45,000 platelets, thrombocytes. So vast majority of the components of your blood or cellular components of blood due to red blood cells. And I've mentioned their primary roles, so again, just emphasizing this here. Let's talk a little bit more about the characteristics. I told you that they lack a nucleus, but they also lack an endoplasmic reticulum, and they lack mitochondria. So I have under here, impact of this question mark. So this is something that I'm going to pose to you now, and then when we meet the next time, I want to see what you guys can think. What is the impact of red blood cells lacking a nucleus, lacking endoplasmic reticulum, and lacking mitochondria. Try to think about this from a functional standpoint. Try not to use outside resources. Don't cheat. Don't Google it. Just try to think about it, and then we'll come to class and we'll discuss what the possible impacts are. All right, so I mentioned that they're also biconcave discs. I haven't mentioned that these red blood cells tend to be very, very flexible compared to other cells. And this is because they're floating, being transported within the blood. Blood is a very dynamic component. Velocity can change, there's shear stress, so they're bouncing off other red blood cells, they're bouncing off the vessel walls. Because of that, they have to be flexible, otherwise they would break apart, and that would be bad. And then the last point here, it says packed with hemoglobin. I don't know the number off the top of my head. I could be incorrect here, but I think it's either 250,000 or 250 million. That's a big disparity, but I know it's 250 something. Around 250,000, 250 million units of hemoglobin per red blood cell. The point I'm trying to make is a red blood cell is basically a dumb nucleus lacking cell that is specialized to transport a lot of hemoglobin. Okay, so let's talk a little bit about hemoglobin. Some basics of it, and that'll lead us into iron transport, and then we'll finish up with problems associated with low red blood cells. So hemoglobin, it's a protein, and it has 4 subunits, 2 alpha chains, 2 beta chains, and at the center of each you have, I don't know why I'm crossing those out, I should just be circling them, but within each chain you have what's called a heme group, or a porphyrin ring, okay, which is being shown right here. At the center of a heme group, or porphyrin ring, you have an atom of iron. So one hemoglobin molecule has 4 heme groups. Another way to think about this is 4 molecules of iron. That's what Fe is. Okay, so what this means is that one molecule of hemoglobin can bind 4 molecules of oxygen or 4 molecules of carbon dioxide, because here at the iron-containing heme group is where O2 or CO2 actually binds. Okay. So hemoglobin, it is, like I said earlier, it's the major component of red blood cells. We've discussed it's that large protein with 4 subunits. Each subunit has an iron-containing heme group. Because the heme groups contain iron within them, what this means is that red blood cells are the major, shouldn't say source or site of iron, our body's iron levels in general. So 70% of all iron in our body is contained within red blood cells. I mentioned the alpha and beta chains, but there can be gamma and delta chains of hemoglobin. For example, I believe fetal hemoglobin, I believe it might be delta, I can't remember off the top of my head, but the exact isoform of hemoglobin changes from that transition between a fetus and postnatal state. And the isoforms can actually slightly differ in humans as in adults as well. I believe some adults have gamma hemoglobin, so it's not just set in stone alpha and beta. However, let's revisit this idea of iron related to hemoglobin. So iron's in the middle of each heme group. We need iron so that we need proper amounts of iron so that we can synthesize the necessary amount of hemoglobin, necessary amount of heme for the hemoglobin for all of our red blood cells. That's kind of clunky to say. Moral of the story, iron's really important. So our iron intake, our iron transport, and regulation of iron levels in our body is very, very important. Iron homeostasis is very important. Most of the iron we get from our diet. So we eat food that contains iron, and it's broken down. I shouldn't say broken down, but it is absorbed in our digestive tract. That digestive tract puts it in the bloodstream, where transferrin, which is a protein, picks it up and transports it in the plasma. So transferrin is a transport protein. Okay. It transports ferrin, which is one of the Latin forms for iron. In addition, our cells can store excess iron in the form of something called ferritin. We join iron up with additional protein, and that combination is called ferritin. This is mostly in cells of the liver. Our liver is a good site of excess iron storage. However, we have a limit to the amount of iron that we can store, and because of that, excess iron can be very, very dangerous. In fact, it can be toxic to both adults, but especially children. In fact, iron overdoses of iron due to accidental intake of iron supplements in children, I believe, is the #1 leading cause of overdose deaths in children in the United States, and I think it's across the world. So still is a common problem. Make a long story short, iron levels are critical to our health. So let's look at some specifics about how it is transported, absorbed, transported, and regulated in our body. So I mentioned earlier, we take in iron from our diet, and that gets transported in our digestive system, and then in our small intestine. That iron gets absorbed across the enterocytes by a specific iron transport process that we're not going into details here. But iron gets transported into the blood, where it's picked up by that transporter that I just mentioned, transferrin. And transferrin's job is to take that iron and deliver it to the bone marrow, because this is the site of hematopoiesis. This is where our body is going to make new red blood cells, make heme so we can make red blood cells. So transferrin delivers iron to the bone marrow, uses that iron to make hemoglobin as part of red blood cell synthesis. Okay? And then a little fun fact here, red blood cells live for about 3 to 4 months, roughly 100 to 120 days in the blood. And because we have a lot of iron and other cellular components devoted to red blood cells, our body is pretty good at recycling the components. Okay, so our spleen actually takes dying or old red blood cells, and it converts that hemoglobin, some of that hemoglobin, to bilirubin, and that bilirubin can be transported to the liver for where it gets combined with bile, and then we excrete it. So that's one way we get rid of, or we can reduce, iron levels in our body. Another possible outcome for unused or excess iron in the form of hemoglobin and that bilirubin is transported to the kidneys, and we can excrete. It's a small amount, but we can excrete some bilirubin in our urine. In fact, that's why your urine is typically colored yellow, is because bilirubin is the one that provides that color. And then lastly, I mentioned if you have excess iron, we can store some of that excess iron in the liver, but there's a finite amount that we can store. That's why liver iron overdoses are potentially fatal, because it causes acute liver failure, acute liver toxicity. It can be very dangerous. All right, so let's see if I missed any information on red blood cells, hemoglobin, and iron transport. I mentioned that we basically recycle elements of red blood cells, not just the heme and the bilirubin, but or heme, I should say. We also recycle some of the other cellular components. Let's see, liver incorporates bilirubin into bile, excretes it via the feces, and also the kidney helps process bilirubin, which gives rise to the yellow-colored urine. Now, here's a clinical aspect or clinical fact associated with iron transport and recycling of red blood cells, and you probably have heard of this. The condition is called hyperbilirubinemia, which is difficult to say, but this is basically a situation where one's bilirubin is elevated above normal for sustained amounts of time. Since hyperbilirubinemia is difficult to say, it's more commonly known as jaundice. Okay? And jaundice can occur in both infants and adults. And in infants, it's actually pretty common because once a baby is born, they actually go through this process where their fetal hemoglobin is recycled. It actually is destroyed and recycled. And because their livers may or may not be functioning fully when they're born, that fetal hemoglobin can accumulate, and then therefore their skin can appear yellow, as well as the whites of their eyes. It can be yellowish in tinge, in color. That's what jaundice is. Good news is very easily treatable by a type of light therapy. It just helps break down the bilirubin so that the babies can excrete it. In adults, if one comes down with jaundice or hyperbilirubinemia, then it's typically associated with liver dysfunction and or maybe liver disease or liver destruction, cirrhosis in the case of alcoholics. So here's a picture of infant jaundice that I pulled from the internet, and I just thought this top photo up here was a good indicator of how easily recognizable jaundice is in infants. Note the coloration of the adult here compared to the infant. It just looks yellowish, kind of orangish, and sometimes if the accumulation of bilirubin is really high, then the whites of the eyes become yellow as well. Okay, so in an effort to keep this video lecture on the short side, I'm actually going to stop it right there, and I'll leave you with looking into the information here in my slides, but also in the book, about disorders associated with red blood cells and all the various causes of anemia. And we'll pick up in class discussing that, and we will finish discussing platelets and homeostasis and coagulation, which is going to be the most challenging part of this chapter. All right, see you in class.